I've been at Clemson for I think like 14 or 15 years. Like it's been long enough I can't even remember for sure. But I've just been all my my professor time has been at Clemson, and I teach bioinformatics, which is using computers to process DNA sequence data, like think like 23andMe, some of the things I'll talk about. I teach undergrads, train students, train PhDs in my lab, and all that kind of stuff. And so, what I want to talk about today is a little bit about just sort of DNA, a little bit of background. I'm not sure what everybody's background is in here. I'm not going to assume anything. So if you've taken high school biology, it might try to tickle some of those neurons a little bit, and then sort of build up to talking about this concept that actually we have a celebrity in the audience, Bill Passman, of being able to really use some of the new technology. And some of this is like Star Trek technology that exists today to help out patients with their conditions, and I'll get into what that means. So to get this started, I'll show this slide again at the end, but we'll put it at the beginning as well. That there's the patient to me. Let me get this going here. The patient is the person that is the most important person in the doctor's office. It's not the doctor, the nurse, the administrator, the insurance collector, the person selling pharmaceuticals, the rep, all those people. It's about the patient and making the patient get better. And our medical system is unbelievable. One of the best in the world, but we could always add more tech and do a better job with this. And so right now, here's a patient. We'll call him Bill, who has a problem. He presented his problem to some doctors. Had some work done and found out that he had a tumor. And in Bill's case, he had a kidney tumor. And was working with an oncologist and has had therapy for the tumor. He's now in remission, and hopefully it'll never arise again. But he still has a tumor type of cancer that doesn't have a treatment for it. And so Bill's a person who needs some help, some options in case this tumor comes back again. And people who get this tumor for the first time, maybe it was caught a little bit later, we can have some therapies for that person. And so there's the concept here of this talk is really bringing scientists into the mix and bringing out new techniques and new technology, new research, and getting it closer to the patient than what was possible in the past, and really centering the patient as the center of the research and treatment activities, right? Because that's what it's all about. And I am an academic. I talk a lot, and so I would like for you to stop me and ask me questions at any point. And if we don't finish the presentation, I just showed you the last slide. So it's okay to do that. This stuff is very complicated, and so please ask questions. I understand how hard all this is, even for me. This stuff I've been working on stuff for a long time. I'm still like, what the heck does that mean? I don't think I understand it. But so really, what I am is it's kind of like a new kind of biologist. I call myself a computational biologist, and I use computers to do a lot of my work. And we work with, I used to actually grow sorghum in the field, do some farming and process it for bioenergy production and do genetics and things like that on there. And I don't do a lot of that kind of work now, but I work with people who work with human tissue and agricultural products from crops and things like that. And so I'm a computational biologist, and I study really the DNA and the output of DNA from plants and animals. I've been lucky enough in my life to have a really weird path to be able to see both the agricultural and the biomedical side of things. I love this stuff. Unfortunately, you can't see it, these molecules, and so there has to be special techniques to be able to see this stuff. I can't even use a microscope for the kind of molecules we're looking at. So I use supercomputers. There's techniques to get sequences of DNA, and you can use supercomputers to be able to process it. I love what I do. But so what we're doing is we're processing DNA, and I'll go a little more detail with this. And we're right now actively working in the areas of trying to understand cancer and how DNA changes in tumors, and also DNA differences in people can lead to different kinds of tumors. We're looking at autism and trying to understand some of the DNA differences associated with autism. We're also looking at beans, like beans that you eat, and the roots, how they recruit bacteria into the roots so that they can make nitrogen, take nitrogen from the air and turn it into fertilizer. So you don't have to fertilize the plants. They can self-fertilize. I'm also working with some former people, colleagues on some low gravity genomics, like where people have grown plants on the space station and trying to understand how they grow and looking at gene expression on those kind of plants. Also with light, how that light affects them. This is really important stuff. If we want to grow plants on the moon, you know, we got to be able to see how they grow in gravity, because they grew in Earth's gravity. That's how they evolved, and that's really important to figure out. And all the work that I do, I have to use these computers, and so I would do a lot of work in what a term called cyber infrastructure, and that's really just a fancy word for like using big computers and writing software and getting people together to make it all work so that you can actually find out a lot of these things we're trying to understand. This is a little picture of my lab. And so my lab is I'm blessed with having people from all sorts of different disciplines. I can't do like just traditional biology, understand just biology. I have to understand lots of different things. And so I work with lots of people who are biochemists. That's actually how I was trained formally. Geneticists, a lot of people who do IT. I mean, like IT in, you know, any organization, helping us be able to process data. Computer scientists, data scientists, which is a new breed of computer scientists, really. Computer engineers, cloud computing architects, bioengineers. I think I saw a bioengineer in the crowd in my lab who's here, Reed. And lots of students in my lab. They're undergraduates, master's students, and PhD students. And I work with a lot of faculty people from around the country. But we study, these are plants and animals that I've studied, either now or in the past. I study plants and animals and then just develop this cyber infrastructure stuff to be able to get the job done. And I'll show you this slide and see if there's any questions, but these are just some of the projects that we do in the lab. And I'll talk about this one in kind of detail. But you've probably all heard about artificial intelligence and how it's changing the world, all that kind of stuff. And we're using artificial intelligence as one of the techniques that we use to be able to learn from data, like find patterns in data, find patterns in tumors, find patterns in people with autisms, their genetic underpinnings to see if there's developmental differences in their brain. Understanding using AI to learn things that are just too complicated for us to learn. Like people are really good at seeing patterns. AI can be more quantitative and see patterns that maybe we can't even notice and go through a lot more patterns. Like if you want to look at pictures, right? You could look through somebody's photo album. Might take an hour to go through a couple albums. The computer can go through it in milliseconds, right? And look for patterns to see, hey, that's grandma, or that's, you know, a picture from Mexico or whatever. It can find patterns to understand what those things are. And so we're using this for a thing called precision medicine. And this is really the heart of where I think things are going, is that precision medicine means to get medicines that are tailored for a person, like use other people's, like tumors, for example, like say kidney cancer, like get a bunch of kidney cancer tumors and look at the patterns in them in gene expression or the way they look under the microscope, and then use those patterns to then see if another person with kidney cancer, if their patterns are a little bit different from everybody else's, or what are the subtle differences. In the case of a tumor, if you know the subtle differences in the DNA, you can derive new treatments for that person that are geared towards that one person, and not a clinical trial with thousands of people, but you can actually develop therapies on an individual basis. And I think this has the power to really just to completely change the quality of life and life expectancy for a lot of people if we can get this down to where we can do this on a routine basis. And this is, we can do it now. The technology is here, but getting good at it is something we're all working on. So this is one of the things we're working on. This is, we're studying these plants, trying to understand how they self-fertilize. And I have a company called Praxis AI that a lot of the computational training that we do, like learning how to program and learning how to look at DNA sequences and stuff like that, we're putting into a training platform that I actually use at Clemson for my training. I'm using it right now in a class called Medical Bioinformatics, and the students are learning a lot of really cool stuff and that I think they can use in the workforce. So with that, I'll stop for a second. Does anybody have any questions about anything?
So I have a question.
Yeah.
And that is, of all these areas of research, what is the most unexpected thing that you've learned? That is, you wound up going in, everybody goes into research with preconceptions. And one of the reasons you do it is to go on and kind of strip those away and understand what the real issues are. What have you learned here?
I think the biggest thing I've learned is, that's a really great question. It's a hard one. That everything is complex. Like as a scientist, you're always trying to chop things up into little pieces and then change one thing and see how the difference is in it. But that's not the way the world works. The world works as complex systems. I mean, like the weather. The weather isn't about the rainfall in Clemson, South Carolina. It's about the weather moving into South Carolina, right? If we're looking at it from here. So looking at complex systems. And so like with genes and genomes, there's thousands and thousands of genes, and understanding how they combine together to give rise to a tumor or to, make roots, recruit soil bacteria into their roots and have a relationship with them. That's what I've learned is this complexity is really how things work. So I'm actually, as I get a little bit older, I'm trying not to chop things up. I'm trying to look at things more holistically and trying to understand how they work. And the other thing that is really cool is that I can write computer code to analyze DNA sequences, and I can use it on your data, Bill, or I can use it on the same computer code on data from plants. It's all DNA. You process it the same way. Any other questions?
I had a question.
Yeah.
I'm wondering, when you're talking about looking for patterns in the genome, are you looking at the entire genome of a person or just certain chromosomes or sequences that you've thought in the past might be associated with that kind of tumor?
Yeah, that's a great question. This is kind of where I'm going to with what we're talking about is that, so you have everybody has, 23andMe. And I'm sure most people have heard of the company 23andMe. What does 23 stand for? Throw a question back at you.
For your chromosomes, the number of
20
chromosomes?
is 23 pairs of chromosomes, right? So everybody's got chromosomes 1 through 22, and then you have XX or XY. So XX, you're female, XY, you're male. And so those are in all your cells in your body. So your genome is all those chromosomes and all the genes that make you a human, or make you a mouse, or whatever your particular organism you happen to be. That those are all over the place. And then the chromosomes are turned, parts of them are turned on and off depending upon the tissue. So parts of the genes are turned on in the brain, parts of the genes are turned on in the foot, in the skin of the foot, or in the liver. And then parts of the genes are that are normally in the brain turned on, maybe they got turned on a little bit differently, a different pattern that gave rise to, say, a brain tumor or something like that. What we do now is you can go and this technology didn't exist really 15 years ago, but you can go and get a sample, a biological sample, and you can look at the DNA and you can sequence the DNA from that sample and see, like, you could sequence like part of the brain that's normal or part of the brain that's not. It's probably better to think about like skin. If you had melanoma, you could get a biopsy of the tumor or the normal skin next to it and compare the DNA differences to it, but also the gene expression differences. We can do this now without having to look at one thing. We can look at everything, and then we can drill down and look at one gene, one thing that we already maybe have some idea how it works. But I look at things holistically as the global patterns of how things are different. If that makes any sense. So what I was going to talk about here is that I'll give just go back into a little bit what a gene and a genome is, because even like I know a lot of people, some people I've noticed that even like getting degrees in biology don't necessarily know this in a deep way. But I'll talk about this as a little bit just misexpression of genes and disease, and I'll talk about the computers a little bit, not too much, and then go into talking about some of this medicine for the individual patient. I like to start off this with this. This is the periodic table, and you've all seen this at some point. Maybe you love it, maybe you hate it, maybe you barely remember it, maybe you know it in detail. But this is all the stuff that makes the planet Earth, right? This is what it's made up of, all these different elements. And organisms on Earth are made up of these core elements. There's the other ones too, but these are sort of the most important ones for building who we are. Carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. CHNOPS is the abbreviation for this. And so of all the things that were created when our solar system was born, these are the things that gave rise to life. So you put these together and then you get organisms, right, for this. This you might have seen before. This is DNA. This is the DNA double helix. And so this is a picture, it's rotating down here. You can see how it's like a spiral staircase. And what this is this is a storage device, just like you have a, you know, a Blu-ray disc storing a movie, or a disk drive in your computer. It's all the information is stored here. So this is a little tiny piece of DNA of the double helix here. And this is made up of, the DNA is made up of CHNOPS without the sulfur. The carbon, nitrogen, oxygen, hydrogen, and phosphorus. These are the elements that make up DNA. And inside the double helix, you have these bases, T, A, C, and G. These are the abbreviations for them. Where A is a shorthand for it's a chemical called adenine, T is thymine, G is guanine, and C is cytosine. And this is really important if you want to understand how DNA works. But just to understand it, you can kind of just think of it as like a 4-letter alphabet, A, T, G, and C. Like we have, what, 26 letters in the English alphabet, right? DNA only needs 4. And what it can do is it can string these together in a sequence, and then the cells know how to read that sequence and then make that sequence turn into something physical that can make the cells, you know, turn into a brain cell, come together with other brain cells, and, you know, be a brain, an organ that can all form an organism and all that kind of stuff. So A, T, G's, and C's. So I like to put this into context here, that if you have a book, right, like here's the Bible, that in the Bible, there's information in the Bible, right? You can read the Bible and you can get information out of it. There's 774,746 words in the Bible, according to the internet. I didn't actually go through and count them myself. But there's 26 letters that make up the alphabet, that make up these words. And so here's a passage from the Bible that, you know, that there's information if you read the passage. And if you go through and make a change in it, like for example, SON could be turned, you know, one change, a mutation, if you will, we're talking about like DNA, goes from sun to sun. So the meaning of this book has changed, this passage. One letter change, that can be a big difference, right? Like this can be like, you know, you go to church and read this, everything's great, but here, you know, you might be a heretic talking about the sun god or something like that, right? And get in trouble. This is a picture of the book that is us. And all of us, if you go through, there's a time during the cells are dividing that your DNA looks like this. These are called chromosomes. And so this is really the book of who you are, and all the information of who you are. Everybody on this call was expressed, you know, you start off as a little egg and sperm fertilized together, and years have gone by and turned you into you, but all the information is in this book. This book, your genome, where these are the different chromosomes, right? 23andMe, 23 pairs of chromosomes, chromosomes 1 through 22, XX female plus XY is male. There's 60,650 words in this genome, in this book. And this is what we know today. And these number of words, we didn't know all these words, you know, 20 years ago. We knew a lot of them. Now we know most of the words. There's probably a few we've missed, but we have kind of the whole book, so we can understand how that book, how that leads to different phenotypes or different traits. 4 letters, right? A, T, G, and C, as opposed to 26 in the English alphabet. This has 6.4 billion letters in it. So there's a lot of A, T, G's, and C's in a genome that you can look for differences in. Notice that there's 2 chromosome 1s. There's 2 chromosome 2s. One came from mom, one came from dad, right? So you have pairs of chromosomes, and that's where you get traits from mom, traits from dad, from these mixtures of these different genes. Everybody on this call has the same set of genes. We all have different flavors of those genes. It might be little minor differences that change the gene expression to make us different, or major differences, right? Like you could have an X chromosome, 2 X's or 1 X and a Y, and this can be very, very different. You can see male and female are very different in development. So you can change these things and the book, and you can change one letter. And for example, this is a real change here. This is a sequence that came from the human genome. This is on chromosome 17, somewhere, you know, along here. And this is a gene that when you have a change, the T goes to a G, then that you get your DNA gets damaged really easily. Your genome gets scrambled up, and it can form tumors. And this is a really a mutation that's found sometimes in breast and ovarian cancer. that you can see these changes. So when you're looking for like changes that are really important, like we're looking for patterns, this is what I'm talking about, is looking for patterns that are associated, maybe patterns not just of one T, but different changes that are associated with the tumor to try to understand what genes are broken in the case of cancer, and is there a way to go back and fix them. I don't do this work, but there's technology now to actually go in and rewrite this code. It's called CRISPR, and you can actually go through and change the, if you get G's, you can change it back to T's. So there is the possibility of rewriting the genetic code when it goes bad in somebody with you know, like with cancer. And I've seen nothing unethical about doing that, because in somebody, if you have a tumor, to go and inject somebody with something that can rewrite the code and make the tumor die. It's like poisoning the tumor by rewriting the code. Questions?
I'm sorry, I'm dominating the conversation here, but I'm kind of going back though to the original thing that you learned and some of the stuff that you've brought up here so far. One thing you went on and mentioned is that, you know, you've learned in particular, look at this stuff holistically, in particular, not going on and breaking it down into parts, but that's an awful lot of stuff to look at holistically without going on and breaking it into pieces. If there isn't a mechanism of doing separation, you know, an abstraction mechanism which to go on and understand this, what are you kind of left with your insight then? Do you go on and have machines understand it for you? Or are you coming up with a different mechanism of dissecting it that you think will help? Or, you know, how do you reconcile your insight?
So so first of all the complexity is there. So if you have a very complex system like the weather, like let's just go to something a little less, you know, so everybody knows. If I go and I look at go to one place, you know, in South Carolina, like go to one street and see if it's raining or not, then I can tell you if it's raining or not. And I've isolated and looked at that one place, but that doesn't tell me about the rest of the place, right? So I need to look at the weather and what's going to change, right? What's coming down the pipe for the weather. That's the way to look to understand what's going on. If I look at that one place, but I don't look anywhere else, if I, you know, close my eyes and focus on that one thing, that doesn't mean the complexity is not there. The complexity is there. And so really what I'm trying to do is accepting the fact that there's complexity and looking at the whole system of all the genes that are expressed at the same time, all those 60,000 words, and trying to understand what they're happening. Because I could reduce it down where I feel better about myself and I can understand if things have changed between a tumor or not, but I'm not really seeing the big picture. I always think of the analogy, you might have heard of this, the toenail of the elephant. So if I think about an African oasis and you go and, you know, you zoom and you see a toenail and you're trying to understand the oasis from the toenail of an elephant, you zoom out and you see it's a whole oasis with lots of animals and lots of elephants and lots of different things. And there's a whole ecosystem going on. But if I don't look at the whole thing, I'll never understand what's going on. If I don't look at it from a high level, holistic level. So it makes things harder, but I think that a lot of times what we're not figuring out is because we're looking too close to the problem. We need to pull out and see the bigger picture. And I cannot, my brain cannot handle this. I need computers and I need artificial intelligence and statistics and things like that to be able to see the patterns. Is that okay? Fair answer?
That's great, thank you.
And this is a paradigm shift. I mean, everybody's trained even people who don't become scientists are trained to be reductionist people and chop everything up into bits. And that is a very, very valid way of trying to understand a problem. But if the problem cannot be reassembled from the bits, you'll never understand the problem. And hopefully I'm wrong and chopping up the bits works because that's easier. Do it that way. And so here's another picture of the genome, and just to kind of go through this a little bit. Here's the genome. So if you have DNA, these DNA sequences I showed you that one, they're very, very long. What I showed you was very, very short. Each one of these like rungs in the double helical ladder is an A, T, G, or C pair. The A always goes with T and G goes with C. And so you can see that this is like, zooming out, and this is wrapped up really, really tightly into these giant these structures here. And what a gene is just an interval. It's just a segment of the double helix. And we have 23 pairs of chromosomes, right? And so all along those pairs, there's just little intervals that store the information of what a gene is. And that's what a gene is, just an interval in the double helix. And all this is, these are pictures of actual chromosomes here. This is still one of the most beautiful things to me, because this is a product of all the evolution that's occurred since whenever life got started. And nobody's ever going to know how life got started unless somebody invents a time machine, goes back and sees it. But when life got started, there was DNA, and DNA changed, and over time it caused all these species to occur. This is an archive, our genome, of what happened. There's still genes in here that we can see are very similar in sequence to bacteria. And so it's all locked in. The history of the human, whatever has led to the human organism, all the organisms led to us, all the generations this time began, is locked into this. So it's like a little time compressed information storage device. It's really, really cool. Again, I've read way too much science fiction and seen too many science fiction movies in my life, just to let you know. So a gene is an interval of the double helix of DNA that it gets copied into a molecule called RNA, and RNA can go and do its function, like lead to traits that which you can see in people, and make different kinds of cells and stuff like that. And a genome is all the chromosomes with all the genes. So it's the whole package of everything. This is looking at a genome here from humans. And so the way this works is DNA. This is DNA here. And I won't go into the details of this, but there's different parts of DNA. Like there's some parts of DNA that maintained, that get through the process of having information in here. And the DNA gets turned into RNA and gets turned into proteins. So DNA making RNA making proteins is sort of the way information flows from the genome to make traits happen. So these proteins are actually the things that go and make the traits happen. You can change the number of proteins and their shape and all this kind of stuff, and you get different phenotypes. In cancer, a lot of times you can get everything the cancer cell is making came from your normal process, right? For processes for that particular tissue, for that kidney cell or whatever. But sometimes the number of proteins change, or the shape of them. You actually get mutations that change the shape of the protein, or maybe the proteins just get shut down and not produced. So you can look at the DNA level and see differences between, say, normal and tumors, and try to find out what's going on at the protein level. And so this is like a real picture of a gene. So this is DNA, A, T, Gs, and Cs. And this is a double helix in real life. We only draw one of the strands of the helix. We just infer the other one. So this just tells you the sequence of a gene. This is the sequence of an RNA molecule that was made from the gene. That was transcribed from the gene. And the only difference really between RNA and DNA is that you have A, C, and Gs in RNA, G, C, A, but you also, the T's get replaced with U. It's just a different molecule that's an RNA that replaces the T. And this RNA can then get translated, and this is a sequence of the monomers that make up proteins. So these are all made up of those, CHNOPS, right, that I talked about before. Those atoms all bonded together. And you get this sequence of the proteins, and these proteins make three-dimensional shapes. So this is a shape of a protein here. So it's really cool to me is that this shape here, this is what's really important, is it has a shape and has a like positive and negative charges, has a chemistry on the surface. And these things can do things like, chop up, the food that you eat, right, into smaller pieces so you can digest it and all that kind of stuff. But this three-dimensional shape is stored in the double helix. And I think it's really cool that it's over all the evolutionary time that made this, this three-dimensional thing is locked into a one-dimensional thing, the DNA double helix. This is showing you that the chromosome is really a storage device. So this is the DNA double helix here in the center. This is a protein that reads the double helix that makes RNA. This is an RNA molecule. So the way you access the information of DNA is you go and read it with these proteins. This is called an RNA polymerase, but it's really the thing that knows how to take the sequence of DNA and transcribe it into RNA. This then can be, you do this for all the different words in the genome, and you get things like a flower, right? There's DNA in the flower that gets produced, all the genes get expressed to get a flower. This is a disc, a Blu-ray disc. And so all the information on the Blu-ray disc can be read not by proteins like it is in the cell. This is reading the information with a laser. But still, this information is here. The images and sounds can get read by this and then turned into, say, a movie, right? So the same thing, I look at it the same way, as these are just storage devices. What's really cool, or the fact that we have computers makes it possible, is that we can read these things like a computer, all the different sequences, and understand what they mean, and have the computer be able to do that, read it fast enough for us. Just like I can't look at this with a microscope Blu-ray, I have to have the spins really fast and laser reads it to make the movie. But it's the same principle. This is the genome. Just to let you know, the genome is about the way it's stored. If you sequence a genome fully, you can store it at about 3.2 gigabytes of data, and a DVD player or DVD can hold about 4.7 gigabytes. And Blu-ray, a single layer Blu-ray, can hold about 27 gigabytes. So you can think about, like, you can put a genome on like a DVD, right? You can, for people who have been around for a while, like, it's better quality than VHS and all that kind of stuff. And you can think about that. So you can store a genome on a DVD. Any questions? I'm going really slow. So I think this is a really good example to kind of show how important this stuff is, or something that's relatable. So if you've gotten Ancestry.com or 23andMe done your sequencing, then they give you a report about how your DNA differences, your ATGs and Cs in different genes, how they can have impacts on your phenotype, how you how you're different for different traits. Like, for example, caffeine processing. So when you drink coffee, you get caffeine in your bloodstream, and that caffeine can stimulate a lot of, the caffeine can touch different parts of your body and stimulate you, right? Can make you more awake. But when you have drugs in your body, be it caffeine or ibuprofen or acetaminophen or a chemotherapeutic or whatever, your liver will go through and grab these molecules and chop them up in little bits. And so caffeine, there's known genes that go and chop up caffeine that are in your liver. And if we know what the gene is, we can know what the sequence is, and we can actually look in the sequence of some of these genes and see if there's patterns that are associated with how fast you chop up caffeine. So if you have this pattern right here, an A and an A in this gene, this is really part of a gene sequence, then you process caffeine really quickly, so coffee doesn't affect you as much. However, if you have two chromosomes, right? One from mom and one from dad. If you have a C and an A at this position, so one of the A's got turned into a C, or you have a C and a C at this position, then you don't process caffeine very quickly. It stays in your blood longer, so it works better. And so you can actually look at your DNA and see stuff like how well you process caffeine, how well you process drugs. And so these are really important genes, like knowing these sequencing for how fast you process drugs that they give you for medical purposes, right? So there's for personalized medicine, for precision medicine, excuse me, knowing these sequences can be very valuable for the physician to give you the right dosage. Some people die because they overdose them. On average, most people don't die from it. You might be sensitive to something. And so knowing this can be very effective in giving you the right dosage of things. Excuse me. I imagine that as DNA sequencing gets faster, and it's going to get a lot faster with things like COVID, right? We need to be able to sequence things more quickly. And the technology is really amazing what's happening, that I can see going to the emergency room, getting your genome sequenced, so they know how much drugs to give you in an emergency situation. They can process it fast enough. Or they go to the cloud, the computer cloud, and pull down your genome. That's your medical record, right? And see how they can dose you. So this is stuff that really is impacting lives. Some of these are being used right now, and I think it'll be used on a routine basis at some point in the near future. So drugs, this is caffeine. here, this is the structure of caffeine, and I kind of want to pull that up really quick. That this caffeine molecule, there's one right there. This is a protein where it's stuck. So all these drugs, what they do is they stick, they interact, they touch the biochemistry of your cells. And I just want to pull this up as a real protein. This is actually protein sequences. Here, zoom is always in the way. But so there's a caffeine molecule right here that's bound to the protein. Well, I'll show you what the protein actually looks like in a second, but this is what's happening. This caffeine sticks to proteins. It sticks to receptors to be able to have a function. If it didn't stick to anything in your body, it would just go through your body, and you'd filter it out eventually. But for it to be bioactive, it's got to interact with your body, just like any drug would. And I think I can just do this real quick. And so this is not perfect here, but these are a lot of the atoms that make up this protein, right? We're looking at the different colors we're in the caffeine. So this actually is stuck inside the protein and bound to it. This actually is a photograph you can take with X-rays. You can shoot X-rays through protein crystals and be able to actually see the atoms. So there are ways to actually visualize these molecules that are inside you that are products of gene expression. This is a structure of DNA, right? So there's the double helix. You can see it as you rotate. And inside here are the different bases inside the double helix. So here's the sequence of this particular double helix. So there's a C, you can highlight right there, a cytosine, right? And there's the cytosine molecule. And there is, C's always pair with G's. So there's a G right there. And so this G goes with the C. I'm just going to click on them so you can see both of them at the same time. And as you scroll down the molecule, you can see the sequence here of the molecule. So there's C, G, C, G, A, T, T, C, G, C, G. As you go through, and there's 3.2 billion in and from mom, 3.2 billion from dad sequences in the chromosomes, right? So this is only a very small number, but you can imagine how big these chromosomes are. So I think I'll kind of I'm going really slow here. I'm going to kind of skip. Well, I'll do this. I'll go through this. So there are some things that are happening now. So because we can do DNA sequencing, we can then go and see the differences in a person or in a person's tumor. You could actually have two different tumors in your life and sequence those tumors from the same organ, and they might be different. They've actually changed, and if you know those differences, then you can start to act on it and do like design drugs and stuff. So there's been there's a big conceptual transition that's happening that happens sometimes in science. It's happening right now, and this is very much a Star Trek type transition, I think. And I like to look at this as an example of one from the past. Is that this used to be the way people thought that humans got reproduced. This is a image from back in 1695. And so this is a sperm cell, and inside the sperm cell, there was the idea that there was a little tiny homunculus, a little tiny person inside the sperm, that the male, of course, the female is not important. The male delivers the sperm to the female, and then all the human comes from the male. And then the woman develops the male's offspring in her womb and all that kind of stuff. This is not a very accurate view of reproduction, right? Of human reproduction. Fast forward a few centuries, and this is a picture from 1953 that was hand drawn by a guy named Francis Crick. So Watson and Crick were two people that were involved with the discovery of the DNA molecule. And so this is a picture of DNA, and this is a drawing that Crick wrote to his son before he published the results. It says the model looks much nicer than this. And I love this because I teach and I draw on whiteboards, pictures of DNA, and my DNA looks better than the person who discovered it, even though I'm a terrible artist. So I think this is really cool. But this is a radical difference, right, of thinking about the world in a ridiculous kind of way, honestly, to really being scientific and understanding what's happening. And so not only now do we understand that DNA transfers information from generation to generation, we know that the DNA can change inside a kidney cells, get mutated, the sequences changes, and you wind up getting a tumor. So that sequencing individual people is causing this other conceptual transition in medicine. And this is the concept of personalized medicine. So it used to be what you would do, like in this particular picture, if you had a bunch of people with a disease like colon cancer, you would get a bunch of people with colon cancer together and try to find a drug to treat them all, right? And so people would try to find the silver bullet that would work for everybody. What happened though in clinical trials, where you're being very scientific and careful, is you would see that some people respond to the drug, they get better. There's a positive effect to the drug. Some people, they don't do anything, nothing happens to them at all. And some people get worse. And so you balance this, these effects and try to find the right drug that works in most people, and you don't kill anybody while you're making the situation worse, right? So you can imagine how many drugs have been tested and didn't work because they weren't good enough, and they didn't work in everybody. So what's happening now is, now that we know we can sequence people's DNA, we can start to group people and start to find people that are in this group, and this group, and then instead of one clinical trial to find the one silver bullet to cure everybody, we can do 3 clinical trials and try and get them where they all have positive effects. And so that's what it's showing here, this personalized medicine perspective. This says future medicine, but this is happening now. That you can do this and find the right therapy for the right people. I would even extend this a little bit, that we're always trying to find as humans the one silver bullet that kills everything or fixes the whole problem. It's probably more of a Chinese medicine type concept of having cocktails of drugs that you give, not one big drug that has all these bad side effects, but lots of little drugs that hit different drug target or different gene targets, and and make the person better with at least a smaller number of side effects. So the complexity of attacking the pattern, essentially. So this is a huge difference. This is probably more important than the concept of this is as important as discovering DNA is what's happening right now. This is a picture of where genes are developed over time. So when you have genes that are misexpressed, you have to go back and try to find out when it happened. This is actually all of us, we don't have this baby picture. It would be kind of cool if we all did, but we all had a baby picture where we're 2 cells, and we eventually went through these developmental stages, and we are who we are today. But these are just different time periods during pregnancy where different genes get turned on that turn on different organs. So if you're trying to understand things like heart development disorders, or maybe even some differences in these organs that maybe happened in the womb, that then stayed dormant for a while, but then give you a higher chance of having cancer. You can look in these time periods, or trying to understand where these time periods are, where to look to find the problems of gene misexpression. And so again, the genes that are all the genes you have are in your fertilized egg when you start off. We're all one cell zygotes at some point in our lives, and these zygotes go through lots of different changes, or as the cells divide, and you get really positive outcomes, right? You get really cool people that get born, right? For example, and you get negative outcomes too, that the genes can be misexpressed, and you wind up getting people that, they're humans and everything, but maybe they need to be censored a little bit more. So we're just people that aren't as cool. But the reason I'm showing this is, over time, you wind up, and all of us over time wind up who we are, but you also have over time things like your cells. There might be mutations in your cells, like maybe in kidney, that you detect at a certain time period, right? You might be 60 years old when you have a problem, you go to the doctor and they go and find that you have a tumor, and you don't know between this time period, the fertilized egg and sperm, first the zygote, to 60 years later in the kidney, when the problems happen. So trying to detect these problems early sometimes can be very challenging, right? Because we don't like to be biopsied if we're not sick. We go and get biopsied when we have a problem and try to understand what's happening. But when this misexpression happens, it can be very very challenging thing to try to understand, see at the right time. And I am definitely, I kind of want to make, I'm going to skip through a couple of things here. These are just databases that we can go and get data from. There's our tax dollars are working very well for getting tumors sequenced and normal tissue sequenced from organ donors. And we actually can look inside the molecular profiles of individual people now in different tissues. Like, for example, this project has, 13 brain tissues have been dissected from organ donors. And then we've looked at gene expression patterns in those tissues. So we can start to understand how genes are working together in combinations to give rise to, the frontal cortex or the cerebellum or the liver, all the different tissues. And again, I'm going to go ahead and, so I'm going so slow, I'm going to skip through some of this, but I have to use a lot of computers to be able to process things. And at Clemson, we have the Palmetto Cluster supercomputer. And these are some colleagues here who are installing a new computer that we use in the lab. And this is a picture up here of the actual Palmetto supercomputer. So we have these computers are, massive. This one has, it's like, this one's 2,000 computers wired together as, as one giant computer to be able to process things. And we use lots of computers around the world to be able to do our work. And a lot of the reason why there's so much data now that we can mine, why we can get, organ donor data from different tissues. The sequencing DNA used to be very expensive. The human genome could have cost, upwards of $1 billion to make. And now we can, use a 23andMe. They have a sale, Black Friday sales for, $100 to get your genome sequenced. Fragmented, but it's still your genome sequenced. And so the cost of sequencing has gotten so low that now it's very possible for anybody to get sequenced if you have some money. Insurance companies are covering this for some people now. So this is a medical record now, it's having your genome sequenced. And this is changing things, right? This is showing you the medical storage system from when I was a teenager, and now medical records, you need supercomputers to be able to store genomic sequences because they're so big. And this is a big opportunity and challenge for doing this kind of work. Again, I'm kind of skipping forward here, but this is sort of just to give you an idea. I think of the stuff that's possible now. It is possible. The tech is here now, and implementing it is happening. People are trying to do this kind of stuff. But I see partnerships now where you can do things like have a company say sequence your DNA, say 23andMe, or maybe LabCorp, right, that does your cholesterol test. They sequence your genome. This information for your genome can be moved around, and there's like networks that can move data around really more quickly than the internet does now. There's like other internets out there using like cloud computing technology, like from Amazon Web Service, for example. Having all this stuff go through to the pharmacy and actually give you the right dosage. So instead of trying to give you a dose of the drug, that your doctor's doing this, right? The doctor's saying you need the statin drug because you have high cholesterol, but actually using your genome sequence to go process it and all this to find the right patterns and associate those with the right dosage. So you can give people the least amount of drug that they need to make it cheaper and make it more effective, and maybe have less adverse drug reactions. So I see these kind of partnerships in the future being very, very important for medicine to be able to dose you correctly and give you the right drugs. Maybe even have your tumor sequence plugged into the system and tell you which combination of drugs to take to get rid of the tumor. And so I'm definitely going way too slow. I'm going to kind of go through this a little bit more quickly here. But we've been using artificial intelligence techniques that some of these are based on the like deep fake technologies. If you've seen the pictures of like Abraham Lincoln animated, like there's technologies to go and take images and then learn how the patterns in them and be able to actually make them move, right? And to understand how to change them over time. And so we've been using some of these techniques, like Colin Targonsky's here, who did this work actually, and Reed Bender, I think, is here too, that they would use some of these techniques to be able to learn how to trick like one tissue to look like another tissue, the gene expression level. So for example, you can go use this technique to go and say, like, take a hippocampus tissue, the gene expression patterns there, and you can trick the artificial intelligence neural network algorithm to go and look like a different tissue. So when you do that, you actually are kind of simulating the differences that change, the expression differences between one tissue and another, find out what genes changed, and then have an idea of what you would need to change to convert between one tissue and another. We've applied this to kidney cancer and other cancers as well, where you can actually go through and take all the gene expression patterns for, say, a normal kidney, and then change them to different, these are different kidney subtypes here, and then find out what the algorithm the computer program told you are the genes that changed. So we've actually applied this algorithm, so it's like going from a normal kidney, using the algorithm to go find out what the tumorous kidney looked like. So again, back to Bill Passman, who's on this call, who's been just a pioneer in trying and getting people to be able to do this precision medicine. That we apply this to his tumor. There's a picture of it right here where he had this tumor DNA sequenced and the RNA sequenced from his tumor. And we're able to identify genes that were different for him in Bill's tumor. We were able to do this because we could take all the government funded tumor samples that exist and use those to like learn what the patterns could be, and then see how Bill's are different relative to these other patterns. So we can find the subtle differences in Bill's tumor profile. And Bill works with one of the most amazing people I've ever met in my entire life, James Shee, who's an oncologist in St. Louis, and he's working with an oncologist who does clinical trials and understands this concept of precision medicine, and can prescribe FDA-approved drugs. Maybe they're approved for different purposes, but they might hit the same pathways that Bill has and be able to, devise therapeutics this way. And so Bill has, I'm sure he'd be more than happy to go into more detail about this, but he's created this rarekidneycancer.org site to help other people with kidney cancers, and not just kidney cancer too. He works with other people for doing this kind of precision medicine in different systems. And Bill and others have been organizing what are called hackathons. This is one that we went to in 2018. I went to in 2018. Another one, I think, from right when COVID was really starting to hit. These are some students from Clemson and some people from the private sector here doing a hackathon, where you go and you bring this data to these people, and to a bunch of people who like have really cool technology and ideas and no biases, and maybe they're not even biologists, and try to find the patterns in the data and try to find ways of treating people. And so this is a early stages, I think, in the history of future medicine, that this will happen, at LabCorp or, at 23andMe or whatever, that this kind of stuff that's happening right now, this having lots of people come together for one person, that it'll be built into the computers to be able to process that, find out the patterns for the individual person. So going back to the slide from the beginning, that this is the patient, and doing this work, working with scientists and using this kind of technology right now is possible. And this Bill's proven this, right? Bill's a co-author on this paper that we published, understanding this. And hopefully what will happen is the science will get so good that this will just get baked into, some computer program that the 23andMe uses, or CVS or the doctor's office uses, but you're going to have the patient's, DNA sequences to be able to do this kind of work. And you can do it now, and there are ways to do it where it's not ridiculously expensive, but it's finding the right, scientists to be able to work on the data. And that's what a big thing I'm trying to do in my lab is train students with this kind of data so they can do these approaches, and it's very motivating for them too to be able to work on data from a real person. And the training that I do, this is my company, Praxis AI. This is actually is a online course. This is just a picture of this of the screen, but you can go through these different skills and learn how to do these things. I teach you some advanced computational techniques, and also how to process this kind of data. Just real quick, this work is not me. I'm like a small part of this whole thing. These are all people that I work with some people around the world, a lot of people around the country, and we have funding from the National Science Foundation. Cisco has given us a lot of funding for a lot of the computational stuff we do, Clemson, USDA, and the Department of Energy. So I didn't mean to talk this long. I'll shut up now if anybody has any questions.
So I have a question again, Alex. First off, thanks again for all of the help that you and everybody gathered here, Colin, Reed, and everybody that's listened. I think that in fact, this is the beginning of something that's really great. You guys are pioneers in it and helping patients who have unresearched conditions make some headway in finding out what to do. One of the things that has been sort of a bottleneck in the past was getting data. So getting the data to you, making it public, having that, and help with the research. Besides the data, what else can patients like me do to help move the research forward with your participation?
So I've learned almost everything from you, right, with this process, and I'm working with a couple other people right now where I'm trying to do a similar kind of thing. And I think that the real bottleneck, or one of the important things, is really getting the patient. This is my recommendation to the people I'm working with, to hook up with the oncologist. And so if the oncologist is not on board, I need to converse with that person and deal with things like where to get the sequencing done and be able to have the data protected and all that kind of whatever we need to do with it. Because I am not part of the medical establishment. The oncologist is that is the medical establishment. Me having a relationship, try to draw these arrows here, between me and the oncologist is really important. So that's what I'm trying to do right now. If there's a person, I'm trying to meet with their oncologist next week and see if they want to do this.
So let me repeat that. In essence, assembling the team that you see on the slide. If you've got the patient, they bring in the oncologist, you have the scientists, and then in addition, I guess now in general, what's kind of interesting from my perspective is the primary care physician and also the oncologist often usually is interested in clinical care. That is, they want to have a standard of care. If they don't go on and do what the book says, they can actually get sued. Okay.
Right.
On the other hand, and then the outer orbit of where they work is with clinical trials. These are things that have gone through an IRB, have made sure that nobody is harmed in the process of doing the thing, or minimally harmed, or if they are harmed, at least informed. And but then in terms of doing pure research, you wind up having a group that's even sort of further out in the orbit. I would argue then that probably there might be even another box here then. You've got the oncologist, but then again, you have the guy that is the specialist. Max Meng is mine, and James Shih, you met that as too. But then you have the research guys. And again, James is more on the research side, as well as the clinical side. So in terms of the guys that go on and kind of wind up being part of this team, could you go on to describe these squares again? Who would you expect to be doing primarily the research and the disease of the people that you show here?
I mean, I think it's a mixture. I think again, as far as the care goes, the oncologist, once you've reached this stage, and correct me if I'm wrong with this, because this is me understanding a lot of this really from you. But so the oncologist is the person that can understand that, okay, they know what treatments to use. If treatments have been exhausted or they're weak, they can entertain the idea of different treatment targets with approved drugs, that the scientists can point to those approved drugs. The scientists can't initiate the clinical trials or anything. Really, the scientists can just process the data and then provide information back to the oncologist. And I would almost argue, or maybe wonder, that this arrow should even be here, right? That the oncologist should be tapping into the scientists, because the patient doesn't need to go figure out where the scientist is, and it's hard enough, right? You're sick. But being able to, have this interface here, this person has to understand what this person's giving them. Like if I give you these drug targets, and it's not to me, like this like the scientific community, then they can act upon it if they want to, and they can go through all the appropriate channels for making sure they don't get sued, right? And first do no harm kind of stuff. But the idea is that if the scientist can point to FDA approved drugs, that makes it possible for this person to set up a clinical trial, or even maybe in the future, if this stuff starts working out, administer drugs to an individual patient based upon good science with it. But I think right now, and I know James is doing some of this, right? The personalized medicine clinical trials of breaking people up into even though they're treating everybody individually, you have groups of people who should respond in a similar way because they have the same mutation profile.
I think I would go on and make, I do have one belief, and that is I think all your arrows are correct. And the reason is that at least in the model that we're doing with the hackathon stuff, the patient does not, if you view this thing as a football game, the patient's understand the rules of the game. They don't play. I mean, they don't make the plays. They're like cheerleaders or potentially organizers that get the stadium in place so that the players can play. So as a result, you need an organizer, and the organizer needs to know who all is going to be coming to the game, right? So that they can get introduced to each other, that all
Yes.
of the roadblocks that are in place are cleared, and the rest of it. So I think you're for the foreseeable future, the arrows as you've drawn them probably are all going to wind up being there simply because the patient winds up organizing the game in the first place. So yeah.
Right, exactly. And that needs to go away, right? This needs to be part of the normal medical procedure is having to, this needs to go away. Right.
Yeah.
This eventually, it's baked into, in medical school, how you get a personalized therapy down. Right now, though, it's not in the medical books. Medical schools don't have time to even teach a lot of the computational stuff that we're doing.
Yep.
But eventually that should be inside computers that in the cloud that you can do that with. But right now it's a manual analysis, right? That needs to feed back to this person. But from like my experience with you, these three groups are critical in agreeing to try to deal with this complex problem, right? Because this is not straightforward stuff. And this guy's busy, and this guy's sick at the very least.
Okay, I think you summarized it real well. Thanks.
Okay, thanks, Bill. Other questions? So I'll put this up again. This is recorded, but if you go through this, this Bill's, the person, the picture that he's popping up. Bill's awesome, and Bill's taught me so much, my students so much. He's taught a lot of people a lot of things. He's doing a pioneering medical work as a non-MD citizen. This is a great blog that he put up recently on March 14th, kind of going through this process of the hackathons. And if you're sick or somebody who's sick, I would suggest reading this. There's a lot of technical stuff, but it's about finding genes. And I think there's a mechanism that will become more and more common. And there are people trying to even monetize this where they can have hackathons, not to milk more money from the poor patient who's paying all this money for medical bills, but be able to have enough resources to sustain and have more hackathons and help more people out. And so this is a movement that is just getting started, I think, and I'm excited to be part of it on any level. Thank you, Bill. I thank you a lot. You're awesome.
Thank you, guys, and Colin and Reed and all of the other guys here that have helped me out. Thank you again. I appreciate it.